Okay, so we're diving deep into prostate cancer research today, right?
Right.
A lot is happening, and we're going to look at some sources, some pretty interesting ones actually,
Absolutely.
covering both clinical trials and preclinical studies
Yep.
to try to get a better understanding of the current landscape and where things might be headed.
Yeah, and I think it's worth noting that, we've got a lot to cover.
Yeah.
And some of it's pretty complex, but we're going to try to break it down, make it as clear as possible.
For sure. We'll try to make sure that it's clear for everyone following along.
Right.
Let's start with the clinical trials.
Well, sounds good.
And the first one up is, well, an analysis of the EMBI-ARC trial,
Yeah.
which looked at enzalutamide
Uh-huh.
and its impact on PSA levels in patients with high-risk biochemically recurrent prostate cancer.
And just to remind everybody, biochemically recurrent means the cancer has come back,
Right.
but it's not yet visible on scans.
Right.
It's only detectable through a rising PSA level.
Right. And the patients in this particular analysis had a PSA doubling time of 9 months or less. Pretty aggressive.
Yeah, definitely indicates a more aggressive recurrence.
Okay, so EMBARK trial had 3 treatment groups, right?
Yeah.
Enzalutamide combined with leuprolide, leuprolide alone, and then just enzalutamide.
Right.
And this analysis was looking at the PSA responses over time within these groups.
Exactly.
So what did they find at week 25?
Okay, so at 25 weeks, they found that a higher percentage of patients who received enzalutamide achieved undetectable PSA levels.
Right.
89% of those on the combination of enzalutamide and leuprolide.
Right.
And 82% on enzalutamide alone reached that undetectable PSA,
Wow, that's compared to...
compared to 63% of patients who are only receiving leuprolide.
Okay, so big difference.
Yeah, big difference.
With the enzalutamide,
Mhm.
seems like it has a pretty big impact on that early PSA control.
Yeah, and they also looked at treatment suspension. And by week 37, treatment was more frequently suspended in both the enzalutamide combination and the enzalutamide monotherapy groups.
Okay, and why is that?
Well, because of that successful PSA suppression, right?
Got it.
So it indicates a quicker and more effective initial response.
Makes sense.
Yeah.
What happens if the PSA levels start to rise again?
So when that happens, they restart the treatment, right?
Right.
And they found that, again, those enzalutamide-containing regimens
Yeah.
had better outcomes. So 96% of patients in the combination group
Wow.
and 90% in the enzalutamide alone group were able to return to undetectable PSA levels.
Okay.
This is compared to only 73%
Wow.
of those who had initially been on leuprolide alone.
So again, pretty big difference there.
Yeah, definitely suggests a lasting benefit from the enzalutamide, even if treatment needs to be interrupted.
Okay, and is that linked to any longer-term outcomes?
Yeah, so they actually found that achieving an undetectable PSA level after restarting treatment was associated with improved metastasis-free survival across all 3 treatment groups. And this was true regardless of which initial therapy the patient had received.
Interesting.
Yeah, it really underscores the importance of controlling PSA levels in managing this stage of prostate cancer.
Okay, so it seems like this analysis from the EMBARK trial really provides some further evidence for enzalutamide, right?
Yes, absolutely.
In managing this high-risk recurrent prostate cancer.
Yeah,
And highlights the importance of achieving and maintaining those low PSA levels.
a key treatment goal for sure.
Okay, let's move on to another set of clinical trials. This time we're looking at phase 1 studies,
Okay,
evaluating 3 different investigational agents,
right.
AZD2284, AZD2287,
Uh-huh.
and AZD2275,
Okay.
all in patients with metastatic castration-resistant prostate cancer, or mCRPC.
That's right.
And because these are first-in-human studies, the focus is on safety, right?
Yeah, safety and also tolerability, how well the patients tolerate the treatments,
Yeah.
and looking for any early signs of effectiveness.
Right, and just to be clear, this is a non-randomized, open-label study.
Right.
So that means all the participants receive those investigational drugs,
Yep.
and both the researchers and the patients know what's being given.
Exactly.
Now, this trial has Part A and Part B.
Okay.
So let's start with Part A, the imaging phase. What's going on there?
So Part A has 2 main objectives.
Right.
First one is called cold antibody exploration.
Okay, and what's that?
So in this part, they're trying to determine the optimal dose of AZD2287,
Right.
both when it's given alone and when it's given in sequence with AZD2275,
Okay.
which is an unconjugated antibody.
Right.
And the idea here is to see if AZD2275 can actually enhance the distribution of AZD2287 to the tumor sites within the body. So it's like...
So it helps it get where it needs to go.
Exactly.
Okay, interesting.
Yeah.
And then there's the Part A expansion.
Right. So in
Like-
this part,
What are they doing there?
They're using advanced imaging techniques to assess how frequently specific biomarkers are present in these patients with mCRPC.
And what biomarkers are they looking at?
They're specifically looking at prostate-specific membrane antigen, or PSMA,
Okay.
and 6-transmembrane epithelial antigen of prostate 2, or STEAP2.
And why are they looking at those?
Well, they're potential targets for future therapies.
Okay.
So understanding how often they're present
Right.
can help guide the development of treatments that target those specific biomarkers.
So it's like laying the groundwork
Exactly.
for future treatment development.
Exactly.
Okay, and then you said there's a part B, right?
Right. So after the imaging phase, we move on to part B, which is called the therapeutic phase.
Okay, and what's happening in part B?
Part B starts with actinium-225 dose escalation.
Okay.
And the goal here is to determine the safe and tolerable range of doses for AZD2284.
Uh-huh.
And they use the imaging data from Part A to guide that process.
Makes sense. It makes sense.
They're also looking for early indications of whether the drug is having any anti-cancer activity. And just to be clear, actinium-225 is a radioactive isotope.
So this is probably a type of radioligand therapy, right?
Right. It's likely designed to deliver that radiation directly to the cancer cells.
Interesting.
Yeah, and after the dose escalation, there are 2 Part B expansion cohorts.
Okay.
These cohorts will further evaluate the effectiveness of actinium-225 AZD2284
Uh-huh.
at the doses they identified in that dose escalation phase.
Makes sense.
Yeah.
But it's still pretty early, right?
Yeah, it is. It's a phase 1 trial.
Right.
So the data is primarily focused on safety and tolerability
Right.
and those initial insights into the potential activity.
Makes sense. So we'll have to see what happens in the future.
Exactly.
Okay, let's move on to another phase 1 trial, this one involving a drug called Pocenbrodib, or CTT2274.
Okay.
What's interesting about this one?
This one was designed with the help of artificial intelligence.
Oh, wow.
Yeah.
That's pretty cool.
It's a big step forward in drug discovery, using AI to actually design the molecule.
So it's not just analyzing data.
No, it's actually involved in the design process.
That's amazing.
Yeah, it's pretty cool.
What is Pocenbrodib actually targeting?
It targets P300CBP, which are epigenetic regulators.
Okay, and what do those do?
So they control how genes are expressed within cells,
Uh-huh.
and they play a critical role in the growth and survival of cancer cells.
So by inhibiting them,
Right.
it aims to disrupt those processes.
Exactly, trying to disrupt the things that fuel cancer progression.
So a new way of treating prostate cancer
Potentially. Yeah.
by targeting those epigenetic mechanisms.
Exactly.
And this development seems really fast.
Yeah, it reached human trials in less than 30 months,
Wow.
which is a lot faster than traditional drug development.
Yeah, usually takes much longer
Yeah, like 5 to 7 years.
to even get to that stage.
Right. So this really highlights how AI can speed things up.
Absolutely.
It's really impressive.
What are the goals of this phase 1 trial?
So they're primarily evaluating safety and tolerability
Okay.
and any early signs of efficacy. And they're also monitoring how well the drug is engaging its target to see if it's actually working as intended.
Interesting.
So yeah, it's a very interesting development,
Absolutely.
showing the increasing role of AI in cancer treatment.
Okay. Let's shift gears a little bit and talk about the LIBERTAS trial.
Okay.
This one's a phase 3 study, but with a different focus.
Right.
It's focused on patients with metastatic hormone-sensitive prostate cancer,
Right.
or mHSPC,
Yeah, that's right.
and it's looking at treatment de-escalation.
Exactly.
So these are patients who've already had a good initial response to standard combination therapy.
Right.
They're trying to see if they can maintain that good response with less treatment.
Yes. And hopefully reduce side effects in the process.
Okay, so how are they doing that?
So the trial is specifically for patients who achieve an undetectable PSA level below 0.2 nanograms per milliliter after 6 months of androgen deprivation therapy, or ADT,
Right?
combined with apalutamide. So these patients are then randomly assigned to one of 2 groups. Okay.
The standard care group continues to receive both ADT and apalutamide.
Right.
The other group undergoes de-escalation, receiving apalutamide alone,
Okay.
and ADT is given intermittently.
I see. So they're trying to see if they can reduce the amount of ADT.
Exactly, because ADT can have some pretty significant side effects.
Right. Things like hot flashes.
Yeah, hot flashes, fatigue, and sexual dysfunction.
So they're trying to find a balance.
Right, a balance between effectiveness and quality of life.
Right. That makes sense.
Yeah.
What were the initial PSA or responses like?
Well, 71% of the patients achieved that undetectable PSA level after the 6 months.
Okay, so a good initial response.
Yeah, pretty strong response to that combination therapy.
So they're trying to see if they can maintain that with less.
Exactly. Trying to find that sweet spot where
Right.
you get the benefit without all the side effects.
Makes a lot of sense.
Yeah,
And this could really change how mHSPC is managed, right?
absolutely. It could provide some much-needed evidence
Yeah,
to support more personalized treatment and hopefully improve quality of life.
absolutely.
Okay, let's switch over to the preclinical studies.
Okay.
And the first one is looking at a drug called MTX-531.
Right.
What can you tell us about this one?
So MTX-531 is a dual mechanism drug.
Meaning?
It inhibits two key signaling pathways in the cancer cells at the same time.
Good.
The epidermal growth factor receptor, or EGFR,
Okay.
and phosphatidylinositol 3 kinase, or PI3K.
Okay, why those two?
Well, in prostate cancer, especially advanced and castration-resistant forms,
Yeah.
one of the biggest challenges is resistance to treatments like ADT and antiandrogens.
Right, the cancer finds a way around it.
Exactly. The cancer cells activate alternative pathways to keep growing and surviving.
I see.
And PI3K is one of those pathways that's often involved in resistance.
Okay.
And while EGFR hasn't been studied as much in prostate cancer,
Uh-huh.
it has been linked to more aggressive disease.
Okay, so by targeting both.
They're trying to overcome those resistance mechanisms.
Okay, so it's like a two-pronged attack, hitting the cancer from multiple angles.
Right.
And what have they found so far?
Well, preclinical studies, mostly in head and neck cancer models,
Okay.
have shown significant tumor-inhibiting effects.
Those are promising.
Yeah, it suggests that this dual targeting approach could be beneficial for prostate cancer too.
Okay.
But we need more research to confirm that.
Right, of course.
But it's definitely a promising avenue.
Okay, good to know.
Yeah.
Now, what about this telomere 1? What's going on with that one?
Telomere 1 is a small molecule that has two main actions.
Sure.
First, it regulates metal ion homeostasis.
Okay, so what does that mean?
It basically focuses on copper and iron levels in the cells.
Uh-huh.
And by normalizing those levels, it helps to reduce oxidative stress.
Okay, and that's important because...
Oxidative stress can contribute to cancer progression.
I see.
So by reducing it, they're trying to slow down that progression.
Okay.
And the second action of telomere 1 is that it activates telomerase.
And what does that do?
Telomerase is an enzyme that maintains the length of telomeres,
Oh, sure.
structures at the end of chromosomes that are important for genomic stability.
So it's kind of like protecting the cell's DNA.
Yeah, you could think of it that way.
Okay, so it's working on two fronts, reducing stress and protecting DNA.
Exactly.
And what have they seen in prostate cancer models?
So in the lab, using prostate cancer cell lines, telomere 1 showed anti-cancer effects.
Okay.
But more importantly, in animal models with human prostate cancer cells, it resulted in about a 50% reduction in tumor growth.
Wow, that's pretty significant.
Yeah, it's a pretty notable finding.
And that was at both low and high doses.
Yes, both low and high doses of the molecule.
Okay, anything else interesting they found?
Yeah, they also found that it might protect against chemotherapy toxicity.
Really?
Yeah, they gave it in combination with paclitaxel,
Does it
a chemotherapy drug,
Right.
and the telomere 1 at a low dose actually mitigated the toxicity of the paclitaxel.
Interesting. So it might help patients tolerate chemo better?
Exactly, that's the idea.
It's like a 2-for-1.
Yeah, it could potentially have direct anti-cancer activity
Yeah.
and also protect against side effects.
That would be huge.
Yeah, it's a very promising candidate for further research.
Absolutely.
Yeah.
Okay, last but not least, let's talk about TROP2.
Okay.
And this is not a drug, right?
No, it's a protein.
And they're looking at it as a therapeutic target.
That's right.
Why is it so important in prostate cancer?
TROP2, which stands for trophoblast cell surface antigen 2,
Okay.
is often overexpressed in a variety of cancers, including mCRPC,
Okay.
and that overexpression is linked to more aggressive tumors and poorer outcomes.
So not good.
No, not good.
And it also seems to be involved in treatment resistance.
Right. It seems to drive cancer progression,
Okay.
promotes cell proliferation, survival, and metastasis.
So it's a bad actor.
Yeah, pretty much.
How are they trying to target it?
The most advanced approach is using antibody-drug conjugates, or ADCs.
Okay, and how do those work?
So they have a monoclonal antibody that specifically binds to TROP2 on cancer cells,
Uh-huh.
and this antibody is linked to a chemotherapy drug.
I see.
So it delivers the chemo directly to the cancer cells that express TROP2.
So it's like a guided missile.
Yeah, exactly.
It delivers the chemo right where it needs to go.
And hopefully minimizes damage to healthy tissues.
That's the goal.
Right.
Okay, and are there any specific ADCs being looked at for prostate cancer?
Yeah, sacituzumab govitecan, or SG, is one example.
Okay.
It's already approved for some other cancers, and
No.
now they're investigating it for prostate cancer.
Okay, and how effective are these ADCs?
Well, it seems to depend on the level of TROP2 expression.
So the more TROP2.
The better it might work.
Okay, interesting.
Yeah.
And you said TROP2 can also be used as a biomarker.
Right, it can be used to identify circulating tumor cells
Okay.
from a blood sample.
So they don't need to do a biopsy.
Potentially.
Yes.
That'd be good. Yeah, it could be very useful for diagnosis and monitoring treatment response.
Okay, so it's a multifaceted target.
Exactly.
They're trying to hit it from different angles.
Right.
Are there any other strategies being explored with TROP2?
There's some evidence suggesting that combining TROP2 targeted therapies with other treatments,
Okay.
like PRP inhibitors or immunotherapies,
Right.
could enhance their effectiveness.
Okay, so it's like a team effort.
Yeah, using multiple therapies to attack the cancer.
Makes sense.
Yeah.
But are there any challenges with targeting TROP2?
Yeah, there are a few.
Like what?
Well, ADCs can have side effects.
Right, because they're still delivering chemo.
Exactly.
Okay, what else?
Also, the level of TROP2 expression can vary
Okay.
between different tumors and even within the same tumor.
So that could make it tricky.
Yeah, it could make it hard to predict how well a therapy will work.
Okay, so there's still work to be done.
Definitely. But it's a very promising target.
Absolutely.
There's a lot of potential there.
Okay, so we've covered a lot of ground here today.
We have.
From enzalutamide to AI-designed drugs to TROP2.
A lot of interesting research going on.
It's exciting to see all this progress being made.
Absolutely.
And hopefully it will lead to better treatments for patients.
That's the ultimate goal.
Definitely.
For sure.
And as always, we encourage everyone to explore the original sources
Yes, definitely.
and related research to learn more.
Absolutely.
Thanks for joining us today.
It was my pleasure.